Hello guys, welcome to our channel Mushrooming Your Knowledge. Today we are going to discuss about an interesting topic in biology, that is information transfer. The name information transfer itself carry some information about our video, that is the process of transfer the data from DNA to protein. So the first step is replication, the second one is transcription, and the third one is translation. The first step, replication, means the action of copying or reproducing something. So DNA copy itself to form another strand of DNA. It's replication. The second step is transcription. What happened in transcription? Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, that is mRNA. And the third step is known as translation. Translation is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins. So we can understand that the final product in the whole process is protein. Today in our video we are going to discuss about only DNA replication. You guys can see the basic structure of a DNA, that is the double helical structure containing the 4 nitrogenous base, that is at A means adenine, T means thymine, G means guanine, and C indicates the cytosine. Uh, Before we step into the molecular basis of DNA replication, we have to know some terminologies like what is template strand, what is non-template strand, what is parental strand, and what is offspring. So the transfer of genetic material from parent to offspring and from cell to cell, for transmission to occur, the genetic material must be copied. What must be copied? Wait a minute. The genetic material. So the gene, DNA present within a gene, we can say that. So template strand. What is template strand? The parental strand from which the new offspring is formed. That is the offspring, and the parental strand is known as template strand. So next, during this process known as DNA replication, the original DNA strand are used as template strands, just what I said now, a template strand for the synthesis of new DNA strand. Okay, we first consider, again wait a minute. We first consider bacterial, that is the prokaryotic DNA replication, and examine how DNA replication occurs within a living cells. And then we turn our attention to the unique features of replication of eukaryotic cell. Why we consider bacterial cell first, not eukaryotic cells? Eukaryotic cells are the advanced one, and the process of replication is more complex compared to bacterial cell, that is the prokaryotic cell. So we consider bacterial cell first. The replication of chromosomal DNA occurs very quickly and very accurately. Just note the term, very quickly and very accurately. Within the given time, the cell replicate itself. The cell is wrong terminology, the DNA replicate itself, and the appropriate time in the life of the cell. For this to happen, many cellular proteins play vital role. So what is cellular proteins? The proteins which is present within a cell, that this term we can call it cellular proteins, play vital roles. So now we will examine the mechanism of DNA replication and consider the functions of several proteins involved this process. The double helix is composed of two DNA strands. You guys can see that the structure of DNA, the double helix, the double helical structure, the double okay, wait a second. The double helix is composed of two DNA strands, and the individual building blocks of each strand are nucleotides. So here the term comes nucleotide, and the another term is nucleoside. Nucleoside. There are different nucleotide and side. not only the S star, not only the S, there are many other difference. You will discuss about the difference in our next slide. So let's continue the slide. The nucleotides contain the contain one of four bases, that is the adenine, thymine, guanine and cytosine. So adenine, the abbreviation of adenine is A, thymine is T, guanine is G and the cytosine is I write it here in the lower part, the C. The double stranded structure is held together by base stacking and by hydrogen bonding between the bases in opposite strand. One strand, if one strand is this, okay, let me change the color, and the another strand is blue one, then the A, T, G, C, those are the bases arranged in a stacked manner one by one, and those bases are bond create a bond between the opposite strand. A bind with blue strand bind with the red one, and the G of the blue strand bind with the C of the red strand. A critical feature of the double helix structure is the adenine hydrogen bonds with, just remember always, A binds with T, AT, and the G bind with C, ATGC. This rule, known as ATGC rule, is the basis for the complementary of the base sequence in the double-stranded DNA. So DNA is always double-stranded. There are some exceptional, but in a general account we can tell that DNA is a double-stranded. Then the difference between nucleotide and nucleoside. So what is a nucleoside? Nucleoside is just nothing, that is the, it contain the nucleoside. Nucleoside contain only the nitrogen base with a pentose sugar, and the nucleotide nitrogen base, pentose sugar and a phosphate group. Here the difference, nitrogen base and a pentose sugar, but in nucleotide there are nitrogen base, pentose sugar and a phosphate group. Here's the phosphate group, PO4 minus, we can say that PO4 minus, phosphate group. Nucleoside, it is component of nucleotide. So nucleotide have হচ্ছে nitrogen base and হচ্ছে কি nitrogen base and pentose sugar, but nucleotide, if we add PO4, that is the phosphate group, PO4 minus, so it converted into nucleotide, then nucleoside is the basic structure of nucleotide. The third difference is it is slightly basic in nature. Nucleoside is basic and nucleotide is in somewhat acidic in nature. Example, that is the adenosine, and the nucleotide is thymidyllic acid. Next one is three possible model of DNA replication. We are going to study replication, so we have to know how replication occurred. Scientists in the late 1950s had considered three different mechanisms to explain the net result of DNA replication based on Watson and Crick's idea. The hypothesis was that DNA replication is semi-conservative. Semi-conservative, you guys can guess the meaning of this was semi-conservative. That is the not fully conserved. The some part of the parental strand is conserved and the some new part is processed in this process. So the first one, here's the cursor. The first is referred to as conservative model. According to this hypothesis, both strands of parental DNA remain together follow DNA replication. In this model, the original arrangement of parental strand is completely conserved, while the two newly made daughter strand also remain together following replication. Let's see the picture. That is the conservative type. The parental strand is blue and the daughter strand, fast replication product and fast replication pen. The fast replication, the blue one, that is the parental strand, and the fast replicated product, that is the fully different one, that is yellow. After that, from the blue one, uh, let us consider as a parental strand, and this is the another parental strand. Second replication, and from the blue one we can get the blue one and a yellow one. And from the yellow one we can get two yellow structure. That is the conservative structure. That is the basic structure of the parental DNA is conserved. Here we can get one blue, from that we can get another blue, from the yellow we can get one blue, oh sorry, one yellow and another one is also yellow. Then what comes, the second one is called semi-conservative. Semi-conservative means not fully, semi-conservative model, not fully conserved. This, in this mechanism, the double-stranded DNA is half conserved following the replication process. In other words, the newly made double-stranded DNA contain one parental strand and one daughter strand. Just mind it, one parental strand and one daughter strand. So one parental, that is the conserved one, and the one daughter strand, that is the new one. So not conserved and the new one not fully conserved. So we can say that it is a semi conservative model. Let us see the diagram. Just wait a minute, change the color in color. Semi conservative model, the parental strand is blue, but when it replicates the first replication step, it forms the semi-conservative structure. The parental strand is blue, the blue one strand have blue, and the other one is yellow. And in another structure, and the second one, one strand is blue and the another is yellow. That's the process, and this process is going on. The third one is known as pen. The third one is known as dispersive model, process that segments of parental DNA and newly made DNA are interspersed in both strands following the replication process. What the parental strand pen? The parental strand is that is the parental strand, and the new strand, those are the new strand. Thus dispersive, that means disperse, the blue one is dispersed with yellow one, so we can call it the dispersive one. That's it. Now we come to the DNA replication in prokaryotic organism. We can consider the prokaryotic DNA, that is the genetic material of a prokaryotic organism. It is circular. So just go start our replication process. Then I will show you how it is going on. First, replication process have three steps, that is the initiation, elongation and termination. The whole process don't have any stages. We divide all those just for our better understanding. There are, you can see the three stages I just told you: the initiation, elongation and termination. Initiation means something, initiation, the proper meaning of initiation is something start. So initiation means starting of the replication. Elongation means elongate, it start to elongate, the chain is elongating. And the termination means end. Termination means end or ending of a process, that is known as a termination process. Bacterial DNA replication. What is bacterial DNA replication? We consider the prokaryotic organism. Bacterial is a prokaryotic organism. So we take bacteria E. coli, that is the Escherichia coli. So we take the example, it is a model organism, Escherichia coli. The site on the bacterial chromosome here DNA synthesis begins is known as the origin of replication. Bacterial chromosome have a single origin of replication. Mark that point. Single origin. Bacterial chromosomes have a single origin of replication. Single origin. The synthesis of new daughter strand is initiated within the origin and proceed in both directions. The another important part is in both directions, that is, it is bidirectional around the bacterial chromosome. This means that two replication forks move in opposite direction outward from the origin. A replication fork is the site where the parental DNA strands have separated and new daughter strands are being made. Eventually, these replication forks meet each other on the opposite side of the bacterial chromosome to complete the replication process. Initiation. In this video, I just only discuss about initiation process. For the next other steps like elongation and termination, we made another separate video for your better understanding. In this video, I will just discuss about initiation. So what is initiation? I told you guys the initiation. Initiation means any process which is begin, that is known as initiation. Initiation is the initiation of replication occurs at specific nucleotide. You guys, I think you guys remember the difference between nucleotide and nucleoside. Nucleotide sequence called the origin. Called the origin of replication, where various proteins bind to begin the replication process in equally. Has a single origin of replication, as do most prokaryotes, called Odyssey site. So what is Odyssey site? Origin of a replication within a chromosome, that is Odyssey site. Ori means origin and C means chromosome. Origin of a chromosomal replication, that is called Odyssey site on its chromosome. The region the region of replication is approximately 245 base pair long. Mark that point, 245 base pair long, and is rich in adenine and thymine sequence. ATGC, A always binds with T, so it is rich in AT sequence. A always bind with T in with double bond, and G with C with triple bond. So we have to, or the replication process have to use much lower energy in the for breaking of AT compared to GC. So the replication start where the AT sequence is reach. DNA replication. Wait a minute, I have to change the ink color. Okay, DNA replication is initiated by the binding of DNA A protein. So the first protein is DNA A. Proteins to sequence within the origin known as DNA A box. So here it binds DNA A box, and the name of the protein is DNA A protein sequence. DNA A box sequence. The DNA A box sequence serves as recognition site for the binding of the DNA A proteins. So what DNA A box, in DNA A box, the DNA A protein binds. So DNA A act as the recognition factor, recognize the site for the binding of DNA A proteins. Okay, mark that point. When DNA A proteins are in their ATP bound form, they bind to 5 DNA A boxes in Odyssey, that is the origin of replication in chromosome, to initiate DNA replication. First the 5 DNA A boxes bind in the Odyssey site. DNA A protein also bind to each other to form a complex with the aid of other DNA binding proteins such as HU and IHF. What is HU? HU is a small bacterial histone-like protein. H. So what does it H means? H means the histone-like proteins, and U is U93 strand used for fast isolation. So it is HU. Where's my cursor? That's it. HU. And the next one is IHF. So what is IHF? IHF is the integration host factor, help to redistribution DNA A proteins for proper binding. IHF, the full form of IHF is integration host factor, and HU is, that is the H stand for histone like protein, and U is U93. This causes the DNA to bend around the complex of DNA A proteins and result in the separation of AT rich region. So here it starts the separation of AT rich region. It start to form bifurcating because only two hydrogen bonds forms between A and T base pair. Here I told you previously AT have two base pair and GC have three base pairs. Her three hydrogen bonds occur between G and C. The DNA strands are more easily separated at AT rich region. Then in our next slide we can see that following separation of the AT rich region, okay, let me get the pen. The DNA proteins with the help of DNA C protein. So second one is DNA C protein. The second one is DNA C. First we get the DNA A protein. Second one is DNA C protein. Recruit DNA helicase proteins to this side. Then here it comes DNA helicase proteins. The third one is DNA helicase. Helicase protein. DNA helicase is also known as DNA B protein. We can say the DNA helicase or DNA B. When a DNA helicase encounters a double-stranded region, it breaks the hydrogen bonds between the two strands. Two strands means A and T have double-stranded region. Let's break it. It have only two, and G and C have three bonds. So if we try to break it, we have to use more energy and it consumes less energy. So DNA, that means helicase, helicase break A and T, it have only two bonds. Two DNA helicases being strand separation within the C region and continue to separate the DNA strand beyond the origin. These proteins use the energy from ATP hydrolysis to catalyze the separation of the double-stranded parental DNA. Parental DNA, the parental DNA, you guys can remember before I just marked the double. What is parental DNA? What is offspring? That's it. In E. coli, that is the Escherichia coli, DNA helicase bind to single-stranded DNA and travel along the DNA in 5' to 3' direction to keep the replication for moving. The action of DNA, mark that point, 5' to 3' direction, okay, not 3' to 5' direction, always 5' to 3' direction. The action of DNA helicases promote the movement of two replication forks outward from ORI-C in opposite direction. This initiates the replication of the bacterial chromosome in both directions and even termed as bidirectional replication. Okay, why it is bidirectional? The DNA, the DNA of a bacteria is circular, so when it starts here, it can forward in this direction or in this direction. Okay, that's why it is bidirectional. And just note that point. It is here it form the bubble. Here it form a bubble-like structure. Here it form a bubble-like structure. It is known as replication bubble. Okay, replication bubble. And here the structure of a DNA double helical structure. The parental strand. Okay. The parental strand. This one is the parental strand, and this one is the new strand. In our next video, when we discuss about elongation and termination, we will discuss about what is leading strand, what is lagging strand, what is free nucleotides. You guys can know that what is, you guys just know that what is replication for. And here the helicase just help to break the bonds between two strands. Okay, thank you for watching. Please, if you guys like the content, Please like, share and subscribe. Okay, just ignore my handwriting. Just like, share and subscribe, and share this video with your friends for their better understanding. Thank you.